Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. We're so glad you're here. And hey, before we jump into today's deep dive, could you take just a quick second to like this video and subscribe to our channel? Your support, it really helps us keep the Sand Science Experience going and lets us bring you these fascinating explorations. Okay, so today we're diving into one of biology's, most enduring mysteries, really. Why do individuals, even ones with the exact same genes living in the same place, have such incredibly varied lifespans? I mean, think about it. Even human identical twins, sometimes one lives decades longer than the other. It's not just us, though. It's a really fundamental question in gerontology that's the science of aging. We're trying to understand what actually drives this variability.
It is a huge puzzle, isn't it? Because it kind of defies our simplest assumptions. So this deep dive, we're going to unpack a really fascinating study by Mosley and colleagues, just published in Aging Cell. They tackle this exact question using C. elegans, those tiny transparent worms. What's amazing is even when they're genetically identical, raised under perfectly controlled lab conditions, they show lifespan differences that are, surprisingly comparable to what you see in humans.
Right. So our mission today is basically to explore how scientists are using these super advanced techniques, almost like biological detectives, to peer into these subtle differences that can actually predict how long a creature might live, even when they're technically the same age, chronologically speaking. We're going to really dig into the sources you shared, especially this new Mosley paper. Okay, let's unpack this then. It just sounds so counterintuitive, If the genes are the same and the environment is the same, what else could possibly be causing it? What's driving this variation if it's not the usual suspects?
Yeah, that is the million-dollar question. And, there have been a few main theories kicking around for a while. One idea is it boils down to stochastic differences, basically random molecular noise. Tiny, unpredictable fluctuations inside cells. Think like maybe a protein misfolds slightly differently here, or some random damage accumulates a bit faster there. These small early differences could sort of nudge individuals onto totally different life paths.
So it's almost like a roll of the dice, but happening constantly at a microscopic level, setting a trajectory early on.
Kind of, yeah. And another related idea is about canalization. So the body tries to maintain stability, homeostasis, right? But maybe some individuals get subtly pushed or canalized into specific ways of failing, specific paths of decline, even early on. And C. elegans, these little worms, they're just perfect for studying this. Short lifespan, genetically identical, easy to control in the lab. It lets you isolate these really tricky factors.
Okay, that makes sense. So if you can't just look at genes or environment, how do researchers even start predicting who lives longer? This must be where those biomarkers of aging come in, these sort of measurable clues.
Exactly. Biomarkers. They can be physical things, phenotypic things, or molecular. For C. elegans, we've known about some behavioral ones for a while, like how fast they move, or how quickly they pump their pharynx to eat, or even how many babies they have. These things tend to decline with age, and they drop off faster in the ones that are going to die sooner. But what's really exciting, and what this new study really leverages, are the molecular biomarkers.
Molecular biomarkers, okay. So what did we know about those before this latest paper? What kind of molecular clues were already identified?
Well, that links back to an important earlier study, Kinzer et al., from 2021, which this new work builds right on top of. They found specific molecular reporters inside the worms. Think of them like tiny glowing tags linked to certain gene activities. Some of these, like len4p.gfp and mir243p.gfp, actually glowed brighter in worms that were going to live longer. Pretty neat, huh? Others, like mir240786 to GFP. And even the worm's natural autofluorescence, its own sort of background glow, did the opposite. Dimmer meant shorter life. They were powerful predictors.
And here's where it gets really interesting, right? You mentioned these markers are redundant. They're in different places, but telling the same story.
Exactly. That's a key insight. Despite being expressed in totally different tissues. Lin4p in the back of the intestine, mir243p all over the intestine, autofluorescence in the intestine and uterus, mir240 just in the spermatheca, part of the reproductive system. They all seem to be tapping into some kind of shared, global health state. It strongly suggests there's this organism-wide process controlling aging, and these different markers are just reading out that same underlying signal.
Wow. Okay, so if these molecular clues are all pointing to one common process, the huge question is, what is that process at a deeper level? How did Mosley and colleagues try to figure that out?
Right, so they took this really clever approach using RNA sequencing. It lets you map out all the genes that are active, the whole transcriptome. First, they established what an average aging transcriptome looks like. They sequenced RNA from big batches of worms at different ages from day 2 to day 12 to get a baseline map of gene activity changes over a normal lifespan.
A baseline map. Okay.
Yeah.
Got it. And then how did they use that with the biomarker worms?
That's the really neat part. They use this very sophisticated microfluidic device. It's like a tiny sorting machine for worms. They took worms that were all exactly day 5 old, so mid-adulthood chronologically. Then, using those 4 fluorescent biomarkers, they sorted them into the top 10%, brightest or dimmest, depending on the marker predicting long life, and the bottom 10% predicting short life.
And crucially, this sorting actually worked, right? These groups ended up having genuinely different lifespans.
Oh yeah, big time. The differences in median lifespan between the predicted long and short groups were significant. We're talking anywhere from a 20% difference to, get this, a 137% difference in remaining lifespan, all based on that one measurement at day 5. It's incredibly predictive.
Wow, that's huge. So what was the first big takeaway from comparing the gene expression in these long versus short-lived groups?
Okay, so this leads straight to their first major finding. The gene expression signatures, that whole map of active genes, looks strikingly similar between the long-lived worms and the short-lived worms, regardless of which of the 4 biomarkers they used for sorting. It didn't matter how they picked the long-lived worms. Their gene activity pattern looked similar. Same for the short-lived ones. This really nails down that idea of a common underlying transcriptomic landscape reflecting future lifespan.
Okay, so it's not just about chronological age anymore, how many days they've been alive. It's more about their physiological age, how old they seem biologically at the gene level.
Exactly that. Physiological age versus chronological age. What they found was that this common gene expression difference largely reflected a shift in physiological age. The predicted long-lived worms, even though they were chronologically day 5, they looked transcriptionally younger, more like day 4 worms on average.
Yeah.
But the predicted short-lived worms, also day 5, they looked transcriptionally older, closer to day 5.5 worms. So what's really fascinating here is it suggests the short-lived animals are literally aging faster at the molecular level, even though they're the exact same calendar age.
Okay, so that explains a lot. They're aging at different rates. But was that the whole story? Or did they find something more specific setting these groups apart, something beyond just being older or younger transcriptionally?
This is where it gets even more interesting, and actually a bit unexpected. While a lot of the difference was explained by this physiological age shift, they found this distinct set of genes that consistently mark the difference between long and short-lived worms, independent of that apparent age shift. They call these orthogonal genes. Think of it like these genes don't change much during average aging in the whole population, but their expression levels clearly separated the worms based on their future lifespan, separate from just looking older or younger. This strongly suggests these specific genes might be closer to the cause of the different aging rates, not just the effect, like finding a control switch.
A control switch. That's a really cool idea. So what are these orthogonal genes actually doing? What biological processes are they involved in?
Well, they found these genes were strongly linked to pathways involved in stress response, like the innate immune system and how worms deal with foreign chemicals, the Xenobiotic response. But, and this is really key, they also found a very strong link to the germline, that's the reproductive system. Genes that were turned up higher in the predicted long-lived worms were often associated with muscles, the intestine, but also significantly the germline tissues.
Wow, the germline. And that connects perfectly with what we already know about C. elegans, doesn't it? It sounds crazy, but if you actually remove the worm's germline cells early on, they can live dramatically longer. It's like the body shifts resources away from reproduction towards survival. There's even that theory about reproductive death, where making babies triggers a decline.
Exactly. The germline's role in aging and lifespan variation is a really hot area. And if you connect this to the bigger picture, recent studies, like one from Eder et al., just this year, 2024, they're proposing that maybe a lot of this non-genetic lifespan variability comes from a sort of decoupling, a breakdown in the coordination of gene expression between the germline and the rest of the body, the soma. So finding these orthogonal genes, specifically linked to the germline and predicting lifespan, fits perfectly into that model. It really sharpens the question. Could subtle individual differences in germline soma signaling be setting these different aging rates right from the start?
That's a really powerful hypothesis. Of course, RNA-seq tells you about mRNA levels, which is great, but it's not always a perfect map of protein activity, so I guess more work is needed to nail down the causal links.
Absolutely. It's a snapshot of gene expression. Future experiments could be really telling. For instance, you could look at these biomarkers in worms that are born without a germline, these GLP-1 mutants. If the biomarkers don't predict lifespan in those worms, or predict it differently, that would be strong evidence for the germline's causal role in this variability.
So this deep dive really gives us a profound insight, doesn't it? It suggests that beyond your genes and your environment, your future lifespan might be heavily influenced by your starting physiological state, and potentially by a specific set of genes, possibly related to how your reproductive system talks to the rest of your body, that dictates your personal rate of aging. It's a real reminder that biological destiny isn't just a fixed blueprint. It's dynamic. It's about how your internal systems are running from day one.
Yeah, exactly. Maybe the thought to leave you with is this. Understanding these really subtle early biological differences, these variations between individuals, that could be the absolute key to unlocking why we all age so differently. And maybe, just maybe, finding ways to nudge those trajectories towards healthier, longer lives down the road.
Well, thank you for joining us on this deep dive into the mysteries of lifespan. It was truly fascinating. And if you enjoyed this exploration, please do take that moment to like this video and subscribe to our channel. Your support really does help us continue the SAN Science experience. We hope you've gained some valuable insights today, and we definitely look forward to bringing you more impactful research and preprints in science right here on the SAN Science experience.